Hello students, I am Jyoti Shoran, your biology teacher. Today we are going to start our next chapter in biology. You have completed the two chapters of human physiology, that is respiration, that is gaseous exchange, and the circulation. Now after digestion many nutrients are absorbed by our blood, and most of the nutrients are used up by body, but still some nutrients are such which are not used up by body, and hence removal of those particular unused substances or the harmful substances necessary for our body, and the removal of these harmful toxic substances is termed as excretion. And in this chapter we are going to discuss about the excretion of harmful waste or nitrogenous waste or toxic substances from our body, and their how they are produced and how they are eliminated from our body. Now your chapter is completely related to these concepts. So what are excretion? What is excretion? Excretion is simply the removal of harmful toxic substances from body. It is termed as excretion. Excretion is not related to the removal of undigested food, that is termed as egestion. That is not excretion. Excretion is removal of toxic substances from the body, and it is related to our excretory system. So what are in general excretory waste produced in the body? Then mainly the nitrogenous waste, which are formed from the amino acids, proteins, and the nucleic acid. They are termed as nitrogenous waste, and they are eliminated as the waste materials. Then why these substances are used as waste materials? Because our body, it doesn't have a capability of storing nitrogen in any of the form. If nitrogen it is immediately required for the formation of amino acids, nucleic acids, and proteins, then the body use this nitrogen for the synthesis of proteins and nucleic acids, and if it is not required at that particular stage, then the body remove nitrogen in the form of some wasteful materials. In total, various type of nitrogenous waste are removed by various organisms, which include generally urea, ammonia, and the uric acids. The amphibians and the fishes, generally the aquatic life, they remove the excretory waste in the form of ammonia, and the process is known as ammonotelism, and the organisms are termed as ammonotelic. While birds, reptiles, they excrete the nitrogenous waste in the form of uric acid, and this is termed as uricotelism, and the organisms are known as uricotelic. And mammals and the higher vertebrates, they remove the nitrogenous waste in the form of urea, because urea is the most intoxic form of the nitrogenous waste, and it requires less amount of water for its removal, and the process by which urea is removed is termed as ureotelism. And the organism, that is mammals, they are classically known as uricotelic organisms. Now what are the various organs that are responsible, that are involved in the mechanism of excretion? If we start from the lowermost organism, then the first excretory system, it appears in the platyhelminthes, that involve the flame cells, and after that annelids, they have well developed excretory system in the form of nephridia. In arthropods, some arthropods also having the excretory system in the form of Malpighian tubule, which are finger like projections which are associated to the midgut or the alimentary canal of these arthropods. And mammals and other higher vertebrates, they have specified well developed systems having kidneys that are performing the formation of excretion and osmoregulation. So mammals and higher vertebrates, they have the most advanced excretory system. So let's start with the excretory system in humans. The human excretory system, it is consisting of a pair of kidneys, a pair of ureters, a urinary bladder, and urethra. These four parts completely forms the human excretory system. If we talk about the presence of the kidney, then the kidney, they are present in the lowermost dorsal area of our abdominal cavity, and they are protected by the lowermost parts of the rib cage, that means lowermost ribs of the rib cage. And if the kidneys, they are 5 to 7 centimeter wide, 10 to 12 centimeter they are in length, and 2 to 3 centimeter are their thickness. The kidneys, they emerge or they arise in the form of elongated tube-like structures that are termed as ureters, and the ureters, they carry the urine formed in kidneys to the urinary bladder. Urinary bladder is a totally muscular organ. It is formed of muscles and it stores the urine. As it gets completely filled, then the urine it is passed through the urethra. So these four parts completely make our excretory system. The blood through the blood vessel through which the blood is supplied to the kidneys for the filtration, it is carried about by renal vein, renal artery, and the renal vein it carries the blood from the kidneys to the heart. Now kidney, if we talk about the internal structure of the kidney, and if we cut LS, that is longitudinal section, means the section along its length, then the internal structure of kidney can be revealed easily. The kidney, it is having very a flask like funnel like structure that is termed as pelvis, and the pelvis it results in the formation of certain projections, and the projections of this pelvis, these are termed as calculi. These calculi, they are associated with the number of conical masses present in the kidney, that is conical masses are known as popularly as pyramids, and these pyramids, they are consisting of a structural and functional unit of kidneys that are known as nephrons. The kidney or the innermost portion of the kidneys, it is divided into the two parts. The outermost peripheral regions, it is termed as cortex, while the innermost part where these pyramids are present, it is termed as medulla. So the region of kidney, it is divided into two parts, that is cortex and medulla. The outermost peripheral region is cortex, and the inner part, it is termed as medulla. Now nephrons, they are also termed as structural and functional unit of the kidneys. So what are nephrons? Yeah, how it forms the nephrons. Nephrons, they are formed of two parts that are known as glomerulus and tubular part. Then what is glomerulus? Glomerulus, it is a capillary network of blood vessels, a network formed by capillaries. These capillaries, they arise, they emerge from the artery that is entering the kidney. The arteries entering the nephron, it gets divided into a fine network of capillaries that forms the glomerulus, and the glomerulus, it arises in the form of again a blood vessel that is termed as efferent arteriole, means the artery, the part of artery that is leaving the nephron, it is termed as efferent arteriole, and the efferent arteriole, it gets surrounded around the nephron, the tubular part of the nephron, and it is termed as vasa recta, means the U-shaped network formed by the capillaries around the loop of Henle of the tubular part of nephron is termed as vasa recta. Now tubular part, it is consisting of various parts, out of which the former one is Bowman's capsule. The Bowman's capsule is a conical flask like structure which encloses the glomerulus. The Bowman's capsule, it results in the it opens into continued into the part of a highly convoluted tubule, that is the proximal convoluted tubule. Proximal means the nearmost part to the Bowman's capsule. The part of the tubular region which is nearmost to the proximal to the convoluted tubule or Bowman's capsule, that is termed as PCT or proximal convoluted tubule. The PCT, it ends in the PCT, it is continued into a U-shaped tubule that arises from the PCT and it opens into the DCT. PCT means pistil convoluted tubule. The U-shaped part of this tubular region, it is forming a particular loop that is termed as loop of Henle, and vasa recta, it surrounds this particular loop of Henle that is formed by tubular part of the nephrons. Now the loop of Henle, they open up into a next convoluted tubular part of the kidneys, that is termed as PCT. PCT and distantly DCT, that is the distantly distal convoluted tubule that is distantly placed from the Bowman's capsule. The PCT and DCT, they are convoluted. Convoluted means coiled to increase the surface area for absorption of the nutrients that are filtered in the glomerulus. So, nephrons, they are termed as structural and functional unit of a kidney. Structural because overall structure of a kidney is dependent on the structure of nephrons, and functional unit because ultimately the formation of filtration, it is performed by these nephrons, and the urine formation, it does take place in the nephrons, and the nephron, they are filtering the waste material from the blood and transporting it into a particular duct that open up into the ureters, and finally the removal of excretory waste from the body take place. Now, on the basis of their presence in the kidney or location in the kidney, the nephrons, they can be broadly divided into two parts, that is the cortical and juxtamedullary nephrons. The cortical nephrons, they are generally short length. Short length means the tubular part of that nephrons is short, and the loop of Henle, this part is too much decreased in these type of nephrons, and these nephrons are that's why limited to the cortex only, and these nephrons, they generally lacks vasa recta. Only afferent and efferent arteriole, as you can see in this particular slide, the afferent and efferent arteriole, they are generally present. Juxtamedullary nephrons. Juxtamedullary nephrons are those nephrons whose tubular part is elongated and they are deeply extended in the medulla of the kidneys, and these are having much more elongated and well defined vasa recta. Now, how urine is formed in the kidney? Now, how urine is formed in the nephrons or the kidneys? The urine formation, it involves simply three processes, that is ultrafiltration, reabsorption and secretion. The ultrafiltration or the filtration of the blood, it occurs in the glomerulus or the glomerular part of the nephrons. The glomerulus and the Bowman's capsule, they collectively formed a particular structure that is popularly known as the Malpighian body. Malpighian body means the junction or the part that is formed by glomerulus and the Bowman's capsule. They collectively forms the Malpighian body, and this Malpighian body, it is responsible for the filtration of the blood. In this, the filtration, the afferent arteriole which is carrying the blood to the kidneys for their filtration, then this arteries, they get divided into a fine network of capillaries that is known as glomerulus. So, first time filtration, it does take place through the glomerulus. From the glomerulus or these capillaries, the remaining the plasma and the blood capsules, there is a sole component of blood, all the nutrients, all the proteins, they get filtered from the blood entering the glomerulus. After passing the glomerulus, that particular filtrate, it passes the basement membrane, and finally the epithelial tissue of the Bowman's capsule, means the single solution or the single filtering substance, it is filtered 3 times, first from the glomerulus, second from the basement membrane, and third from the Bowman's capsule's epithelial tissue. So, it make the blood too much filtered that no any impurity and no any substances other than the blood plasma or blood capsule is remained in the blood, and the blood, it is highly filtered. The filtration is too fine, that is, it is termed as ultrafiltration. After ultrafiltration, the filtrate which is having waste and nutrients as well is pumped, is put into the tubular part of the nephrons from the Bowman's capsule. After filtering, the filtrate, it enters the lumen of the Bowman's capsule, and the lumen of Bowman's capsule, it is continued in the form of tubular part of the nephrons that are termed as PCT, loop of Henle and DCT. Now, after ultrafiltration, the reabsorption of the nutrients and the water and ions, they are reabsorbed from the tubular part of the nephrons with the help of vasa recta, and the tubular part, it also secretes some ions and hormones which are regulating the mechanism of urine formation. So now let's start with the mechanism of urine formation, means how urine it is formed in the kidney. The urine it is formed, it start with the ultrafiltration that occur in the glomerular part of the nephron, and after that the tubular part of the nephrons, it plays a very important role in the urine formation. So first of all, PCT is get highly coiled to increase the surface area for resorption and of the ions and the minerals. So in PCT, the most of the ions, nutrients and water, they are absorbed from the filtrate by the cortical fluid or cortical substance of the nephrons. After this resorption, the PCT they opens into the loop of Henle, or simply the Henle's loop. The Henle's loop, it is consisting of two limbs. There is a descending limb which runs downward and the ascending limb which runs upward. Means the descending limb it runs from cortex to medulla, and the ascending limb it runs from medulla to the cortex. Now descending limb it is permeable to water only, and the ascending limb it is permeable to ions only. Means the descending limb it is impermeable to ions and only permeable to water. That's why the water that is present in the ducts which has come from the PCT and finally from the lumen of the Bowman's capsule where the filtrate entered the nephrons, and then from this filtrate maximally water is reabsorbed by the medullary substance of the nephrons, and this absorption of water make this filtrate highly concentrated. Means hypertonic filtrate is formed in the loop of Henle's descending limb. After this highly concentrated filtrate, it enters the ascending limb that is permeable to ions only. Means the ascending limb it is impermeable to water but permeable to ions, and from this particular limb of the loop of Henle, the maximum reabsorption of the nutrient or the ions it does take place, and remaining the urea it is impermeable to urea, and hence the ions including sodium, potassium for example, and some other nutrients involving glucose etc., they are absorbed by the medullary substance to maintain the pH or to maintain the composition of the blood as well as urine constant or preferable. After that, the filtrate it enters to the distal convoluted tubules, that is again the coiled part of the tubule that is placed distantly from the Bowman's capsule. This DCT it also increases the surface area for absorption. In DCT the ions like sodium, potassium and hydrogen, they are secreted and passed to the DCT, and this makes the maximum absorption of sodium ions, chloride ions, carbonate ions and water from the DCT, and the DCT it releases the filtrate into the collecting duct. Collecting duct is a common duct in which the millions of nephrons open, and these collecting duct they open into the calculi, that is the, you can see here, that is the projections of these pelvic region or broad funnel shaped region of the kidney, and these calculi they collectively open in the ureter, and ureter it leads the urine to the urinary bladder from where the urine it is passed out from the body. Now how pH or how the concentration of urine is determined. The concentration of urine it is determined with the help of a well defined mechanism that is termed as countercurrent mechanism for urine formation, and this mechanism it is performed and it is maintained by the arrangement of vasa recta and Henle's loop, whose two parts are opposite in direction to each other. Means this this descending limb of vasa recta, it is opposite in direction to the ascending limb, and similarly the descending limb of the loop of Henle, it contains the direction for flow opposite to the ascending limb of loop of Henle. It makes a permeability difference between two membranes, and the ions and the minerals, they are exchanged between these two membranes, and the pH of the urine, it is determined. In this, the filtrate that is entering the convoluted tubule, it runs downward through the descending limb of the loop of Henle. That is permeable to water only. Now, water, it comes out from the loop of Henle. The vasa recta, ascending limb of the vasa recta, that is closer to the descending limb of the loop of Henle, it is permeable to water, but the water released by this descending limb of loop of Henle is absorbed back by this particular descending limb of vasa recta. Now, the ascending limb of the loop of Henle, it is permeable to ions, and it start to release sodium ions, chlorine ions, other nutrients and some other ions into the metric substance of the medulla. These ions, they are absorbed by the descending as well as ascending limb of the vasa recta. The ascending limb of vasa recta, it is also permeable to the ions and minerals, and these are reabsorbed or absorbed back by this particular ascending limb of vasa recta. Now, the filtrate that is leaving the loop of Henle, which were hypertonic in the descending limb of loop of Henle, is now hypertonic as compared to its concentration in the cortical part of nephron. Now, this permeability difference maintains a concentration gradient, means by exchanging the water, the sodium ions, potassium ions and other nutrients and salts that are present in the blood, by the exchange of these materials, they maintain a particular concentration gradient, and this concentration gradient, it makes the concentration difference across the membranes, and this concentration difference, it is responsible for the flow of certain ions molecules from the membrane that are transported actively as well as passively. For example, the ions such as the ionic larger ions, glucose or the larger molecules, their transport involves energy and it is actively transported by these membranes, while the urea, that is not necessary for these particular membranes and it is transported passively, and the ascending limb of the loop of Henle, it is permeable to urea too, means the some amount of urea, it is absorbed back by the blood so that it can maintain the concentration and of the matrix as well as blood and the urine, and to maintain these particular membrane, this concentration gradient is much more important, that is maintained by the countercurrent mechanism of vasa recta and the Henle's loop or the loop of Henle. After performing this particular mechanism, means when the filtrate, it leaves the ascending limb of loop of Henle through the DCT, that is the distal convoluted tubule, it opens into the collecting tubule. The collecting tubule, its membrane is also permeable to water and urea, means maximum amount of water and a little amount of urea, it is reabsorbed by the inner medullary substances or the matrix, so that the concentration of urine, it can be determined. So, the countercurrent mechanism which is maintained by these particular structures, vasa recta and the tubular part of nephron, by exchanging the materials or ions between them, is responsible for the determination of concentration of urine. Then how urine formation the mechanism of the urine formation is regulated? It is regulated by the two hormones, out of which first is ADH hormone or the antidiuretic hormone. When our body start to lose fluids in excess of amount, then a pH imbalance is generated, and this pH imbalance, it triggers the hypothalamus to release the hormones that is termed as vasopressin or ADH, that is antidiuretic hormone. The antidiuretic hormone, it triggers our osmoreceptors, and at the in the influence of this particular hormone we feel the thirst. This ADH hormone, it triggers the osmoreceptors, and the osmoreceptors, they are start to absorbing these particular ions and also releasing the to absorb the fluid and also start to releasing the ions so as to maintain the pH. As the pH is balanced, there is no signal to the hypothalamus from our body, and when hypothalamus, it doesn't get any signal from any part of body, then it doesn't release its hormone. So the osmoregulation by these osmoreceptors, it acts as negative feedback for the regulation of the for the secretion of ADH hormone. As the pH, it is balanced with the help of osmoreceptors, the hypothalamus, it stops to release the ADH, and also the osmoreceptors, they are also switched off. And in this mechanism, the antidiuretic hormone or ADH hormone, it regulates the urine formation. The ADH or vasopressin hormone, it most preferably act on DCT and the collecting duct, so that the maximum of water can be reabsorbed from the urine that is passing away from the body. Now the second mechanism is renin-angiotensin mechanism. Renin-angiotensin means two enzymes, they are responsible for the regulation of urine formation. It is associated with the GFR. GFR is the glomerulus filtration rate, means how much urine or how much substance is filtered by our glomerulus or the kidneys per day. The GFR, it comes about 125 ml per minute or 180 liters per day. As due to any imbalance in the, which may be due to decreased blood flow or due to any of the reason, malfunctioning of the kidneys, as the glomerular filtration rate, it get decreased, it acts as a signal for the juxtaglomerular apparatus. The juxtaglomerular apparatus, it is formed by the afferent arteriole and the DCT, means the part which enter the glomerulus and the part which leaves the loop of Henle or the tubular part of the nephron, they collectively form the juxtaglomerular apparatus, where DCT and afferent arteriole, they combine, it form JGA or juxtaglomerular apparatus. When the juxtaglomerular apparatus, it receives signal as decreased glomerular filtration rate, it releases renin. This renin releases angiotensinogen, that is inactive, means angiotensinogen that is present in inactive form in the kidneys. The renin, it converts angiotensinogen to angiotensin 1 and angiotensin 2. The angiotensin 2, it is the most important required factor for the secretion of aldosterone by the adrenal gland or the kidneys. The aldosterone released from kidney, it increases the absorption of sodium and potassium ions and water and other nutrients that are lost into the form of urine from body in the distal convoluted tubular part of the nephron. Thus, this increased absorption of the nutrients from the urine, it increases the glomerular filtration rate, because presence of these ions increase the viscosity and it result in the increase of blood pressure. The increased blood pressure, it act as a signal for the atrionatriuretic factors that are A and F. The A and F, they start to release their enzymes or hormones which dilate blood vessels. Dilate blood vessels mean they broaden the lumen of the blood vessel, and these broadened lumens, it result in the decreased blood pressure. And when this blood pressure is decreased, it acts as the normal GFR and there is no signal to JGA, means the decrease in blood pressure after the activity after increased glomerular filtration rate act as a negative feedback for the JGA and the juxtaglomerular apparatus. It stops to produce renin and ultimately without renin the angiotensin secretion is also stopped and finally aldosterone and the GFR it comes to its normal rate. And this mechanism in which renin and angiotensin are involved for the secretion of aldosterone to maintain the glomerular filtration rate is termed as renin angiotensin mechanism for urine formation. And in this way the urine formation it is regulated by our body by the means of some enzymes and hormones, in which antidiuretic hormone and the aldosterone are important. ADH it is secreted from the hypothalamus while aldosterone it is the renal secretion. Now the urine formed it must be passed away from the body, then the passage of urine from body is termed as micturition. The signal from micturition arises from the bladder itself. When the urinary bladder it get completely filled, the walls of the bladder start to stretch, and this stretched wall they act as signals for the central nervous system, and the central nervous system it causes contraction in the urinary wall and it result in the opening of urethral opening and the urethral, after opening of the urethral or urethra, the urine it gets passed away from the body. If we talk about the composition of urine, then urine it generally consists of water, urea and salts. And pH of urine it comes around 6, means a little acidic due to presence of this urea. Color of urine is generally pale yellow due to breakdown of the bilirubin pigment present in the bile. These bilirubin pigment when break they result in the formation of pale color pigment, and then pigment is also present in the urine, that's why the color of urine is yellow. Approx 1.5 liter of urine is released per day, and approx 25 to 30 gram of urea is produced by kidneys per day. And along with kidneys, some other organs also act as the excretory organs, which generally involves skin. The skin it also releases some nitrogenous waste in the form of sweat. Along with water, ions, some urea is also lost in the form of sweat, which forms a alternative method for the urea elimination or the waste elimination or excretion. Sebaceous gland, the sweat gland of the body, which generally secrete wax. Along with this waxy secretion, the sebaceous gland they also eliminate a little amount of urea, but the role of sweat glands and sebaceous glands for the purpose of excretion is negligible. Sweat glands they are modified for the thermoregulation, means to regulate the temperature of the body, and sebaceous gland they are generally involved in the lubrication of our outermost regions or the epithelia. Now lungs, the CO2 it is also a waste product that is formed under the process of respiration. It is also it is also to be removed from the body, and it is done by the lungs with air by exhalation. Then means when we exhale air, then CO2 it is exhaled by lungs or with the help of respiratory system. Now the our modern lifestyle it is resulting in a number of disorders. So our excretory system it is also facing a number of disorders, out of which the most fatal is the kidney failure, means the failing of kidney to filter the blood. And in this case, when the kidneys they are not capable of filtering the blood, then only hope it is the alternative way, that is the hemodialysis. Hemo blood dialysis filter करना means the to filter the blood with an artificial method or with the help of machine. In this, a dialyzer is used to filter the blood. That we what we have to do, an artery of our blood of our body is connected to the dialyzer with the help of some apparatus. When this blood enter the dialyzer, to avoid the coagulation of the blood, an anticoagulant, that is heparin, is pumped into the blood. So heparin, when the blood it reaches the dialyzer it is having a continuous supply of dialyzing fluid that enters from this particular site, means from here fresh dialyzer it enter the solvent, it enter the dialyzer, and from here the used dialyzer it leaves the dialyzer. When the blood pass through this dialyzer, then it is consisting of semi-permeable membrane. When the blood pass through these semi-permeable membrane, the solution or the solvent used in the dialyzer, it is consisting the concentration as same as the plasma and the interrenal fluid, but leaving the urea, means it is lacking urea completely, and our blood it is consisting of urea. When the urea containing blood pass through this urea lacking dialyzer, the urea it generally passed to the dialyzer following the process of simple diffusion, and in this way the blood it get filtered. Now with the help of a particular apparatus, this is connected to our veins, and from vein this filtered blood is carried to the heart, and the heart it pump this filtered blood to whole of our body. When the kidney they get failed completely, then the person it require a regular dialysis, which may be weekly, twice in a week, or within 15 once in 15 days. And if dialyzing is not possible, then the only possible treatment is kidney transplant, means to transplant a kidney from a healthy donor, which is generally his close relative, is into the defected or the affected person, is termed as kidney transplant. Then the second most common disorder of the kidneys is renal calculi. Renal calculi means the formation of insoluble masses of some salts. For example, oxalic acid is forming oxalate. Oxalic acid is formed from urea, and the oxalates they start to deposit in the kidneys, and these are popularly known as kidney stone, which are very painful. The other disorder which may get arise due to any infection or inflammation in the glomerulus. The inflammation it generally occurs due to the microbial infection in the glomerular part of the nephrons, and when the glomerulus they get inflamed or the affected or infected, then they are not capable of filtering the blood properly, and hence our excretory rate or excretory system it is affected adversely or badly. With this I am completing this chapter. Okay, read the chapter carefully. If you are feeling any doubt and any query, then please contact me without any help.